Hi, and welcome to the unit on influenza, which is the flu. So the flu is caused by a virus called the influenza virus, which we're going to focus on this unit, but we're going to use it as a way of talking about um, characteristics of viruses in general, whether viruses are alive or not, and what it means to be alive, evolution and how viruses evolve, how the immune system works, and then we'll finish up talking about vaccines, which are our major defense against viruses apart from the immune system itself. So a virus is a simple particle that typically we don't consider to be alive, but it is infectious. It finds a host cell and will infect it and forces the host cell to reproduce it. So a virus doesn't contain a cell. It only has at minimum two things, a protein shell around it called the capsid, and inside of that capsid, it has genetic material, either DNA or RNA. So whereas a cell has like a membrane and then it has a bunch of cellular machinery inside of it, viruses don't have that. They're very, very tiny. They have this little protein shell and then some genes inside of that. They don't have any metabolism, meaning they don't get energy from their environment. They also have no chemical reactions inside of them of any kind. To go along with that, they have no ability to reproduce. So they rely on host cells to reproduce them, and any energy that's required for that process is going to be provided by the host. So at minimum, I said a virus has a capsid, which is the protein shell, and it has genetic material inside of it. It's possible that viruses could also have other things. Some viruses have enzymes inside the capsid as well. Those enzymes are always inactive. They're only going to activate once the virus infects a host. And inside the host, they would be used for various things. Some of them might be used to help replicate the virus. Some of them are used to help take the host over or overcome host defenses. Another thing a virus might have is an envelope, which is a membrane around the capsid. The envelope, if the virus has it, is actually taken from the membrane of the previous cell, the previous host cell that the virus infected. The point of having an envelope for a virus is that it contains proteins from host cells because it is a bit of membrane from a host cell, and that can help you evade the immune system. So if you get infected by a virus that has an envelope, it'll use one of your cells as host. It'll force that cell to reproduce many, many copies of the virus. Those new viral copies will be released, and they will try to find new host cells inside of your body. Because this is an envelope virus, each one of those viral particles contains a little snatch of cell membrane with proteins from the previous host cell, which was one of your cells. So it has your proteins on it. That means that when your immune system takes a look at those viral particles and it sees your proteins there, it might think that's part of me. So having an envelope will help a virus evade the immune system of its host. On the other hand, it also has drawbacks. It makes it more vulnerable anytime the virus is outside of a host, just in the wider environment, because a membrane is not very strong compared to the protein shell. A virus always has to have proteins on its surface in order to attach to the next host. So if the virus has an envelope, then those attachment proteins need to be on the envelope. And that means that anything that disrupts the envelope membrane will basically get rid of the proteins that the virus needs to attach to the next host. If a enveloped virus loses its envelope, it's not going to be able to infect new host cells anymore. So if you see like Lysol or any other disinfectant product that advertises that it gets rid of viruses, those kinds of products are only getting rid of enveloped viruses, and what they're actually doing is destroying the envelope membrane. Um, that kind of product is never going to work on a virus that doesn't have an envelope. But most viruses that infect humans do have an envelope, including the flu virus, and also the COVID virus. So that brings us to the characteristics of life. Weirdly enough, we actually don't have a single definition for life. Instead, we have lists of characteristics that we say living things have, and if you satisfy every one of those characteristics, then you are considered alive. But actually, different scientists will have different lists. So the characteristics here are some of the most commonly cited. The first is cells. The cell is the basic unit of life, so every living thing has at least one cell. The second is reproduction. Every living thing can reproduce. Third is information. There's two kinds of information involved with organisms. First, organisms have hereditary information, which is information they pass down to their offspring in the form of genes. And second, information that is sensed in the environment. So all organisms have the ability to sense information about the environment and respond to it. Fourth, energy. All living things have the ability to acquire energy from their environment. They also have a metabolism, which is chemical reactions happening in the cell. And a lot of those reactions are used to produce ATP from whatever energy sources are available in the environment. Finally, all living things evolve. So if we compare those characteristics to virus characteristics, we see that viruses are not um, making they're not making the list. So they don't have a cell. They don't reproduce. Instead, they rely on a host cell to replicate them. They do have hereditary information in the form of genes, but they don't sense information in the environment and respond to it. They don't have energy, so they don't get energy from the environment or use it in any way. They have no metabolism at all. And then fifth, evolution, they do evolve. They have genes that they pass on to their descendant viral particles, so therefore they evolve. So that means that most scientists consider viruses to not be alive, although it's obvious they're related to life because they have genes. On the other hand, there's some scientists that say the only thing that's really necessary for life is just evolution. And if you think that, then you would say that viruses are alive. Viruses have two possible quote unquote life cycles or replicative cycles that they can follow, lytic and lysogenic. The lytic cycle is where the virus enters its host cell and pretty much immediately forces the host to build new copies of it, which are then released from the host cell, killing the host. In the lysogenic cycle, the virus will go dormant when it enters the host cell, and after some period of time passes, then it can activate and enter the lytic cycle. So we'll go through these steps of the lytic cycle in more detail. The first step is attachment of the virus to the host cell. This requires surface proteins on the virus. They're going to be located on whatever is the outermost surface for that virus. So if this is a virus that has an envelope, then the attachment proteins are on the envelope. Otherwise, they're going to be on the capsid. The attachment proteins on the virus need to bind to a receptor on the host cell so that the virus can grab the host cell. The receptor on the host cell is not like intended for the virus. It has some other purpose for the host cell. The virus is just using it as its receptor. What kind of receptor the virus can attach to determines what the virus can use as a host, both what species it could use as its host and within that species, what kind of cells, what cell types could it use as host. So there's some viruses that will infect pretty much any one of your cells. There's other viruses that can only infect a few cell types or even just a single cell type. One of the more famous examples of that would be HIV, which basically uses a particular kind of immune cell as its host, rather than any other kind of cell in your body. Once the virus attaches to the host cell, then you have step 2, penetration. The virus has to enter the host cell. Then you have step 3, synthesis of viral components. There's 2 things that need to be made in step 3. First, you have to have viral proteins made, and second, you need to copy the genome of the virus, which is its genetic material. So there is no virus that is able to build protein. That means it's always going to be the host cell building viral proteins with no exceptions. When it comes to copying the genome of the virus or copying its genetic material, sometimes the host cell will be responsible for that. Other times, some of the enzymes needed will actually be provided by the virus. But no matter what, all of the building blocks are coming from the host cell, and all of the energy for the process is also coming from the host. Step 4 is assembly. So you're making new viral particles. You have new capsids forming. Every capsid is going to be packed with the genetic material of the virus. And then step 5 is release of those newly formed viral particles from the host cell, which typically will kill the host. And if this is a virus with an envelope, then as each new viral particle emerges from the host, it's going to take a patch of host cell membrane with it, which is another thing that leads to the host cell dying because it can actually literally run out of membrane. For the lysogenic cycle, we will talk about the steps briefly, but they won't all be on the test. Basically, it's just the cycle where you have dormancy involved. So it starts out the same as the lytic cycle. The virus will attach to its host cell and penetrate it. But then it gets a little bit different. So in step 3, the virus will go dormant inside of its host. Then in step 4, it remains dormant for some period of time, which actually could be lasting for years. In step 5, it reactivates from dormancy, which could be triggered by different things. We're actually not sure all the things that can trigger a dormant virus, to wake up. And then at that point, it re-enters the lytic cycle. So you'll go through synthesis, assembly, and release of new viral particles. Something else to note about lysogenic viruses is that when they're dormant, usually they will actually integrate their genome into the genome of the host cell, which means their DNA will literally become part of the DNA of the host cell. When that happens, the virus is basically permanently a part of that host cell. If the host cell were to divide, then it would copy all of its DNA, and that would include copying the DNA of the virus and passing it on to the offspring of that cell. For their genetic material, viruses can have either DNA or RNA, which is unlike cells. Cells always have DNA that they use to store their hereditary information. But actually, for viruses that infect animals, most of them use RNA instead of DNA as their genetic material. For any virus using RNA as its genetic material, it has a problem when it's trying to have the host cell copy its genome, which is that animal host cells are not able to copy RNA from an RNA template. They can use DNA as a template to build new DNA or to build RNA, but they cannot use RNA as a template for building anything. So that means that an RNA virus needs to have its own enzyme, which is going to be responsible for copying its genome. The building blocks and the energy are provided by the host, but the actual enzyme is coming from the virus. RNA viruses usually will have a really high mutation rate because the enzyme that they use to copy their genome typically is not very good at doing that. It makes a lot of mistakes, and those all end up as mutations. And this is typical both of influenza and the COVID virus, actually. Both of those are RNA viruses. They have their own enzyme that copies their genome, and those enzymes make a lot of mistakes, meaning that you have a really high mutation rate in the virus, and that's why we have new flu strains that evolve every year. That's also why during the COVID pandemic, you had so many new strains of COVID coming out frequently. There's also a special kind of RNA virus called a retrovirus, which has an RNA genome, but then instead of building new RNA copies of it, it actually builds a DNA copy of that genome. And then it puts the DNA copy into the DNA of the host. For humans, the biggest example of that kind of virus is HIV, actually. So HIV has an RNA genome. It makes a DNA copy of its genome when it enters its host cell, and then that DNA ends up incorporated into the DNA of the host cell. And at that point, it'll go dormant for a while until eventually it reactivates later on. And basically, that cell permanently has HIV as part of its genes. The other option for viruses is to have DNA as their genome, which is just like normal cells have. And there are some human viruses that use DNA for their genome. For this kind of virus, it can rely totally on the host to copy it, because the host cell already has enzymes for copying DNA, so it can just copy the virus's DNA too. However, they still have a problem, which is that host cells are not going to actually make the enzyme for copying DNA unless they're preparing to use that enzyme, meaning that they would be preparing to divide and copying their own DNA for that purpose. And of course, most of your cells as an adult are not actually actively dividing. Because your adult cells are not trying to divide, they don't make the enzyme needed to copy DNA. They're not planning on doing that. So a DNA virus, in order to actually successfully replicate inside of a host cell, usually it will need to push that host cell into the cell cycle. That will cause a little overgrowth of cells, like a wart, and can promote cancer as well. So warts typically are just caused by DNA viruses that have forced their host cell to enter the cell cycle and divide. So now that we've gone over the basics of viruses, we'll talk a little bit more about the flu virus and how it causes the flu. So the flu virus is influenza. There's a bunch of different strains of influenza. We name them based on the surface proteins that they have. They have two major surface proteins that are used to attach to host cells and penetrate the host cells, which are H and N. And then for each of those proteins, there's different versions of the protein that we have identified, which we give different numbers. So you're naming flu strains with the letter and number of each protein on its surface. So H1N1 has the first version of the H protein and the first version of the N protein. H5N1 has the fifth version of the H protein and the first version of the N protein. The different protein versions for H and N will determine what receptor the virus prefers to use to enter a host cell. That is going to determine what species the virus can infect and which specific cells in your body it can infect. So the flu is always infecting cells that line the respiratory tract, but the specific cells that each strain of the flu prefers can be located in different parts of the respiratory tract. That's what this diagram is illustrating. For the H1N1 flu, the receptors it prefers are found on cells high up in the respiratory tract, like even just in the nose itself, meaning that if you breathe in some of those viral particles, they find a host cell right away there in the nose. That means that this kind of virus or this kind of flu strain is really easy to transmit, but it usually doesn't cause a serious disease. Whereas for the H5N1 flu strain, it doesn't have any receptors that are located in the nose. It doesn't use the same receptor as the H1N1 strain. The receptor it uses is found down in the lungs. So when you breathe in particles of the H5N1 flu, they need to go all the way down into the lungs before they can find a host cell. Usually that's not going to happen, so it's really hard to transmit that kind of flu. On the other hand, once it does transmit, it's already creating an infection deep in the lungs, which is by definition a serious infection. So that kind of flu, that strain of flu, will end up with a really high death rate. When the flu virus enters the respiratory tract and finds a host cell, it will attach to the host cell and penetrate it using the H and N proteins. Then it follows the lytic cycle, so it immediately forces the host to synthesize viral proteins, and it'll start copying its genome. Once the first host cell dies, it's going to be releasing thousands of new flu particles that'll all go on to find other host cells inside the respiratory tract. The death of that first host cell is also going to activate the immune system. Then the immune system is going to respond with inflammation. And anytime you have inflammation, you're going to have swelling. In the respiratory tract, if you have inflammation there, it's also always going to increase mucus production. So that's what causes the cough and congestion that you have with the flu or the runny nose. If it does get down into your lungs and you end up with the swelling and the inflammation there, that can cause pneumonia. You're also going to have white blood cells, which are immune cells, called to the area. They'll be attracted by the inflammation. When they arrive, they're going to start releasing cytokines, which is just a kind of signaling molecule for the immune system. Cytokines are attracting even more immune cells and activating them. They're also changing the activity of different cells, and they can actually cause a lot of the symptoms of sickness as well. Like they can cause you to lose your appetite and have a headache, and inflammation will cause like aches and pains. So basically this means that most symptoms of the flu are not caused directly by the virus. They are caused by the immune system responding to the virus. They're caused by inflammation and by cytokines. That's why there's so many different diseases that cause flu-like symptoms, including COVID actually caused flu-like symptoms. Basically anything, any kind of pathogen that infects the respiratory system is going to cause the same flu-like symptoms because those are just the symptoms of the immune system turning on in your respiratory tract. We also have vaccines for the flu that come out every year. That's because the virus is constantly evolving and it evolves very quickly. So we always have new strains emerging every year. The way that the vaccine works is by teaching your immune system what the new flu strain looks like, so that if you actually become infected by that strain of the flu, your immune system will already be able to recognize it and will respond really quickly. So that brings us to evolution. We'll talk about evolution in general, and then we'll return to how the flu evolves. So evolution is the change in the genetics of a population over time, or a change in traits over time, but it has to be traits that are caused by genetics. So for any given gene, you'll have different versions of that gene that are possible that give rise to different traits. Like for instance, for eye color, you have genes that control eye color, and there's different versions of those genes which give you the different possibilities for eye color. And different eye colors could become more or less common in the population over time because the gene versions that create them are becoming more or less common in the population over time. So individuals do not evolve, only populations can evolve. Individuals can have mutations that could create new versions of genes, but individuals do not evolve because evolution is specifically about the makeup of a population. If you have a population change a lot, it can actually change so much that it becomes a different species. We call that process speciation, and that's actually where all species come from, meaning that all species come from other species, and therefore all species are related to each other by common descent. So we all share a common ancestor. So this is a super summarized version of the tree of life. It has all major groups of organisms that exist today on it, and at the base of the tree of life, you have the last universal common ancestor that everything that lives today ultimately has descended from. There's multiple processes that drive evolution to occur. The most famous is natural selection, and that's the one that we'll focus on. But there are other things like random changes, actually, and sexual selection, which means like competition for mates or attracting mates. Finally, evolution is the unifying theory of biology. So it is the one theory that holds together all of the knowledge in biology and makes everything make sense. Without evolution, you just have a lot of random facts that it's not clear how they're connected to each other. So before we talk more about where the idea of evolution comes from and evidence for it, we'll talk about how natural selection works. Natural selection is just the idea that if you have advantageous traits, it's easier for you to have offspring and you probably have more offspring. So maybe your advantageous traits make it easier for you to find food or easier to get away from predators. Therefore, you live longer, you have more opportunities to reproduce, and so you have probably more offspring. Then those offspring are going to inherit your traits. As long as those traits are based on your genetics, they will be inherited by your offspring or can be inherited by offspring. So those traits will then become more common in the next generation because of how many offspring you had. On the other hand, if you have disadvantageous traits, it's harder for you to find food, it's harder for you to get away from predators, so you maybe live less long and have fewer offspring, and then those traits are less common in the next generation. The key to natural selection is that what traits are advantageous or disadvantageous depends entirely on the environment, and the environment can change. So this example is moths living in a forest in England, and originally in this particular forest, most of the trees had this light colored bark, and most of the moths are white with black spots, which lets them blend in really well with the bark of the trees. During the day, the moths will like lay flat against the tree and try to hide, and then you have birds that are trying to eat them. And sometimes you have a moth that has a mutation that gives it a dark coloration, which is a disadvantage in this environment because you stick out really clearly against the bark if you are a dark colored moth in that environment. And then the bird comes and eats you, so you probably don't live that long or have that many offspring. So there was never a lot of dark colored moths in the population because they just struggle to live long enough to have offspring, or to have a significant amount of offspring. Then the Industrial Revolution happened, and you actually had a factory built near this forest that was putting out a bunch of air pollution and a bunch of soot, and that soot actually ended up coating the trees. So the color of the trees changed to be dark. Now all of a sudden the environment is different, and which trait is an advantage or not has changed. So now if you're a dark colored moth, you blend in with the trees better. And the white colored moth with black spots is the one who sticks out and has a disadvantage. And that's the one that gets caught by the predator birds. So really quickly after this happened, the population of moths in the forest became dominated by dark-colored moths, whereas the white-colored moths are struggling to have offspring. The dark-colored moths are successfully hiding from the predators. And actually later on, when they eventually got some laws limiting air pollution, the forest did go back to the original coloration of the trees, and the population of moths also switched back. So you can never tell what is going to be an advantage or not. It's all based on the interaction of traits with the environment, and then the environment can change. So you might actually be able to tell what's an advantage like right now, but for the future, you're not able to know what trait is going to be an advantage in the future. So now we'll talk about where the idea of evolution comes from. Um, Before we get into that, we'll mention what people thought about the diversity of life before evolution was thought of or before it was accepted. So people used to think that species do not change over time. The species that we have now are the same species that have always existed on Earth for as long as Earth has been around. And we used to think that species are not related to each other and that they are ranked. At some point, we started ranking them from higher to lower, and of course, we're at the top, and how much you look like a human determines where you rank in the hierarchy of species. Charles Darwin, of course, is the scientist who is credited with the theory of evolution. He wrote a book on the origin of species in 1858, which proposed the idea of evolution and also included a bunch of evidence supporting evolution, most of which he collected during his travels in the Galapagos Islands, where he looked at a bunch of different plant and animal species and also looked at fossils. I made a bunch of drawings and observations that were reproduced in the book. Um, Actually, he didn't write the book in 1858. He published it in 1858. He had already had it written and like completed for a while, but he was hesitating to publish it. He kind of saw it coming that it wasn't going to go over very well. And in 1858, he received a letter from another scientist, Alfred Russel Wallace. Alfred Russel Wallace also independently had the idea for the theory of evolution, and he wasn't sure if he should be trying to publish this idea, so he wrote to Darwin asking for advice. And Darwin told him, yeah, you should publish the idea, and he also took his own advice and published his book. So they ended up both publishing in the same year, 1858. However, it's Darwin who gets the credit for the theory of evolution because Alfred Russel Wallace only published an idea. Charles Darwin had the idea as well as a bunch of evidence to support it. And Charles Darwin is on top here. Alfred Russel Wallace is the guy on the bottom. So Darwin was inspired by a number of different things. Of course, a lot of his inspiration came from the years he spent traveling in the Galapagos Islands. Um, and he noticed a lot of different plant and animal species during those travels, but the most famous thing that he noticed was different finch species. So every island in the Galapagos has its own species of finch living there. And then you also have a different species of finch on the mainland, which is Ecuador. In addition to different finch species on the different islands, you also have different plant species on the different islands, and finches will eat seeds for their diet, And Darwin noticed that all the different finch species have different beak shapes, and it looks like the beak shapes of the finch species are really well matched to the kind of seeds that are most commonly found on the island that finch lives on. So he thought that maybe finches came over from the mainland, from Ecuador, and eventually made it to all the different islands in the Galapagos and found different food sources there when they got there, and over time adapted to be able to eat the different food sources easily. Darwin also had a hobby of breeding pigeons. So we all know what wild pigeons look like, but there's actually a bunch of crazy looking pigeon breeds that you can get. Um, and this just basically proves that like species can change. So if you can go from a normal pigeon to this guy just by picking which pigeons get to have offspring together, obviously species can change. And Darwin basically had the idea like if you can do that deliberately, then a similar process could be happening naturally. He was also inspired by geology. Just like we used to think that species don't change, we also used to think that the surface of the earth doesn't change. And so if you have a canyon in a particular place, that canyon has always been there since the beginning of the world. But they were starting around this time to recognize in geology that that is not the case, that the Earth's surface does change, it just changes really slowly. And that idea was popularized by a geologist, Charles Lyell. So he said that Earth's surface does change over time. The change is very slow. That means that Earth itself is very old. In order to get the surface features that we see, a massive amount of time must have passed. And he also said that if you go somewhere like a canyon and you notice the different layers of rock there, those layers are in order by age. So the layers on the top are young and the layers on the bottom are old. And you had another guy, William Smith, who took that idea about rock layers being in order based on age, and he extended it to the fossils that you find in the rock layers. And he said, if you find fossils in a old layer of rock that's located deep, those fossils are older than the fossils found in the higher layers of rock. And this is a diagram that William Smith made of fossils and rock layers in the part of England where he was living. So he has the different rock layers in that part of England and then the different kinds of fossils that it's typical to see there. So Darwin took a look at this and he just kind of noticed change over time. Darwin was also inspired by the economist Thomas Malthus. Thomas Malthus had the idea that basically it's possible for a population to get bigger than the amount of food available for it. Populations increase exponentially, but food production will only increase linearly. So at some point, you're going to have the population get higher than the food supply. And at that point, you have what was called a Malthusian catastrophe, basically a famine. Darwin just had the idea that if that is applying to humans, then there's no reason why it wouldn't apply to animals and other things living in the wild as well. I'll mention Thomas Malthus's ideas have been basically discredited at this point. Um, the thing that he didn't take into account was technology. If you have a technological advancement that affects food production, then you can pretty much instantly massively increase the amount of food that's produced, which means that food production normally would increase linearly, but sometimes it can increase vertically if you have new technology introduced. And in that way, the food supply can actually keep up with an exponentially growing population, at least for a long time. So those are the sources of Darwin's inspirations for the theory of evolution. we'll talk about the evidence that supports evolution in a moment. First, we'll talk about where genetic diversity comes from, and it comes from mutations. So in order to have evolution, you have to have a population where different individuals have different um, genes. They actually all will have the same genes, but they'll have different versions of each gene. The fancy name for a version of a gene is an allele. So every gene will control one particular trait, and that gene will have different alleles possible, which will give rise to different versions of the trait. For example, for peas, these are some different possible traits of pea plants. One of them is flower color. Pea flowers could be purple or they could be white. So you have a gene that controls the color of pea flowers, and there's two different alleles of that gene, one that gives purple flowers and one that gives white flowers. Same for seeds, you have a gene that controls the seed color. You have two different alleles of the gene, one that gives yellow seeds and one that gives green seeds. So that's the genetic diversity in a population. In order for evolution to happen, you need to have new alleles coming about, and those alleles come from mutations. A mutation is a change in the genetic sequence of a gene. If that change is passed down to future cells, then it's a mutation. Usually we think about mutations as being bad. A lot of the time they are bad for the organisms that have them. Overall, they are good because they're the source of life's diversity. But actually, most mutations are just neutral. Most mutations don't impact the function of a protein in any way, for good or for bad. Then you have some mutations that break the protein that the gene makes. And then occasionally you have mutations that actually give you a version of the protein that works better than before. And very, very rarely you'll have a mutation that gives a protein that has an entirely new function that it didn't used to have. Those kinds of mutations are going to be selected for in natural selection and passed down to offspring. Actually, even for a mutation that breaks the protein, that might not be a bad thing. It could even be a good thing. Again, it depends on the environment. So you have an example here with these two different mice. The mainland mouse lives in like woods. He has a dark coat which matches well with the ground where he lives. The beach mouse, of course, lives on the beach, which is a light color, and he has a light color to match the beach. The coat color is controlled by a protein called the melanocortin receptor. The melanocortin receptor will allow pigment to build up in the fur of the mouse. And what's shown here is a little bit of the DNA of the gene for that protein, and then the mRNA that you get, and then a little bit of the protein itself. So when you're making this protein from the gene, you will take the information in the DNA, copy it into mRNA, and then the mRNA is used to build the protein. The normal sequence of the protein is what's shown here on the left. That gives you a fully functioning melanocortin receptor that will accumulate pigment easily into the mouse's fur, which gives the mouse a dark coat, which for this mainland mouse helps him blend in, so that's great. It's possible to have a mutation to the gene for the melanocortin receptor that breaks that protein. And that's what's shown on the right here. Here you have a mutation that just changes one G to an A. So it's a single base change. That ends up changing the amino acid at that location in the protein from an arginine to a cysteine. And when you have a cysteine here in the melanocortin receptor, it actually breaks the protein. So that's why this mouse is so pale. The melanocortin receptor is working really badly, so it's not able to build up pigment in the fur of the mouse. Even though the protein is broken, though, that's actually not a bad thing for this mouse because he's living in an environment where the ground is pale, so it's good for him to be pale to blend in. If you had this kind of mutation occur in the mainland mouse living in the woods, that would be a disadvantage. That would be a major disadvantage. But if you're living on the beach, then it's advantageous actually. So, whether a mutation is good or bad really depends on the environment. Mutations mostly come about because the cell is copying its DNA and it makes a mistake. It's inevitable that some mistakes are going to be made anytime DNA gets copied. The enzyme that is responsible for copying DNA actually can check its work, and usually when it makes a mistake, it will notice that and like fix it, but you're not going to fix like everything. So any mistake that gets made and never gets fixed is going to get passed on to offspring cells, and that's going to become a permanent mutation. So that's the source of most mutations. The other possible source of mutations is from mutagens. A mutagen is a chemical or physical agent that causes mutations. Most mutations are completely random. And it's important to note that natural selection does not cause mutations. So that means that if the environment changes or if a population moves into a new environment that has different features than the previous environment they were adapted to, maybe they're not well adapted to this new environment, they are not guaranteed to get adaptations that will help them live there. Natural selection will not create those mutations. And any mutations that occur are purely random. So whether they're helpful or not is just up to fate or up to random chance. If the population doesn't happen to get any mutations that randomly help it survive in this new environment, maybe the population is going to just die out. On the other hand, if you do have like one or two individuals in that population that randomly happen to get a mutation that helps them survive there, natural selection will make it so that those mutations become more common in the population. So natural selection does not create mutations. It does not guarantee you adaptations. But if you already have a random mutation that makes you better suited to an environment, natural selection will make it more common. So now we'll talk about some of the evidence that supports evolution. First, you just have the basic fact of extinction, which doesn't necessarily prove that like one species creates another species, but it proves that species change. And actually, scientists literally used to think, and people in general used to think, that it's impossible for something to go extinct. Everything that has existed has always existed, and nothing else has ever existed. It's actually pretty hard to prove that something went extinct. Maybe you haven't seen it in a while, but maybe you just haven't happened to see it in a while. So for a long time, even though some people were suggesting that it's possible for species to go extinct, most scientists were not taking that seriously because it's really hard to prove it. But this guy, Baron Georges Cuvier, proved basically to the satisfaction of scientists at the time in 1812 that it is possible for something to go extinct. And basically all he did was say like, look at the fossils we found of gigantic organisms, like this giant sloth here. We don't see those organisms. We've been to every continent now. We've been to every forest. And sure, there's a lot of stuff that we haven't found, but everything that's like multiple times as large as a man, we've found. You're not going to have gigantic, monstrous creatures that we just haven't seen at all, but we see their bones. So that means that those creatures are actually extinct. And that means other things can go extinct too. Now, actually, scientists estimate that 99% of all species that have ever lived have gone extinct. One of the most important sources of evidence for evolution is the fossil record. So we can observe, of course, fossils of things that don't exist anymore, and we can observe them changing over time. And initially, when Darwin was around, you could just kind of order them based on what rock layer they were found in. But nowadays, we have radiometric dating, and we can actually find out exactly how old different fossils are within a range of error. So, looking at the fossil record, we've been able to see that life has existed on Earth for around 4 billion years. Earth itself is about 4.5 billion years old, so life has existed for most of the history of Earth. And for most of that time, life was nothing but single-celled organisms and prokaryotes. So the first cells to evolve were prokaryotes, they didn't have nuclei. And only later did eukaryotes evolve. Eukaryotes are actually only around 2.5 billion years old, but even when eukaryotes evolved, they were still single-celled organisms. We actually don't see multicellular life emerging in the fossil record until around 1 billion years ago, and those were still very small organisms, microscopic organisms even. Organisms big enough to see with your eyes are only around 500 million years old. So in the fossil record, we're able to see that life has a very long history on Earth, and we're able to see a progression over time from simpler life forms to more complex life forms. And a couple of things you can observe in the fossil record are the law of succession and transitional features. The law of succession is just an observation that a lot of the time you'll see a correspondence between animals that live in a particular area and some fossils that are found there. So for instance, in South America, today you have armadillos living there, and in the fossil record you have the fossil of this guy, which looks quite similar to an armadillo. In Australia, today you have wombats living there, and in the fossil record you see the skeleton of this guy, which looks very similar to a wombat. Transitional features are features that are in the middle between an older trait and a newer trait. And the example here is the transition from fins to limbs. So you start out with the older trait, fins, which have bones in them, and then you go through different transitional forms to the younger trait, which is basically legs. And you can watch the bones change over time to become less fin-like and more leg-like. Unfortunately, there's a lot of gaps in the fossil record, or basically a lot of things missing from the fossil record, because most of the time when something dies, it doesn't make a fossil. In order for something to fossilize, when it dies, it needs to be quickly covered by mud and sediment. So in most places, that's just not going to happen. Another piece of evidence for evolution is vestigial traits. A vestigial trait is a degenerated remnant of an ancestral trait that was actually functional. In humans, we have a couple of vestigial traits. One of them is the tailbone. So this is your tailbone. It is 3 to 5 different vertebrae that never really develop and instead fuse together to this one little tailbone. But of course our ancestors had proper tails. So the tailbone is the remnant of a real tail. It has no function, it's a degenerate trait, but it's basically what's left over of an ancestral trait that had a full function. Wings on an ostrich or another flightless bird are another example of a vestigial trait. They are too small to allow the bird to fly, so they don't have the actual function that they used to have, or that the ancestral, that the ancestor of the ostrich, their wings had. And creepily, Cave fish. When fish live in a cave for a long time, like there's no light there, so they don't actually have a need for their eyes. And it's actually a waste of resources to be like making an eye that you're never going to use to see anything with. So cave fish will inevitably like lose their eyes once they live in a cave for long enough, for enough generations. But they still have the eye socket where the eye used to go as a vestigial trait or a remnant of a trait in an ancestor. We can also make comparisons between living organisms and their structures. And in particular, homologous structures are evidence of evolution. Homologous structures are structures that look similar to each other because they share the same evolutionary origin. They may still have the same function, or they may have different functions actually, but their structures show that they have the same origin. You also have analogous structures, which are different. Analogous structures look similar because they have the same function, but they don't share an evolutionary origin. So this table is showing that if you compare the leg of a cat to the leg of a praying mantis, they have a similar shape. Those are analogous structures. They're both used for walking, that's why they're shaped similarly, but they do not share an evolutionary origin. If you compare the leg parts of a praying mantis to the leg parts of another insect, you see the same parts, even if the legs are used for different things. For instance, the water boatman leg is actually used for swimming. But they have a similar structure because they're both inherited from the same ancestor. For the cat, if you compare the bones of its leg to the bones of any other mammal, you'll identify the same bones. Maybe the shape is different, but the arrangement is the same. And that's because, of course, they share a common ancestor. So there's lots of examples of homologous structures in biology. So these are some of those homologies. There's different types of homologies. The leg bones are an example of a structural homology. You can also see the same thing with arm bones. You can look at the arm bones for any mammal, and here a bird is actually not even a mammal. And you'll identify the same bones in each limb. Maybe the shape is different in different species because the functions can be different. Some of them for walking, or for grabbing, or for flying, or for swimming, but the actual bones are the same, and their arrangement is the same because they share a common ancestor. There's also developmental homologies, which are homologies that occur during embryonic development and may be lost later on in the adult. So, for example, if you look at the embryo of any mammal, you will see little gill slits. Like these little slits here, they look like gills. If you were a fish, those would develop into gills. Of course, humans don't have gills and no mammal has gills, but every mammal as an embryo has the structure that would have developed into gills because we've all evolved from fish. There's also genetic homologies, which are similarities in the genetic code or in the genes between different species. And actually, the genetic code itself is an example of a genetic homology. So the genetic code is how you translate sequences of bases in the DNA into amino acids in a protein. And it's done the exact same way for all life on Earth. From you all the way down to a bacteria, all life shares the same genetic code, which is really weird if we're not actually related. Then if you look at specific genes, we actually share a lot of our genes in common with other life forms as well. Our closest living relative is a chimpanzee, and we have actually over 98% similarity in our genetic sequences with a chimpanzee. So 98% of your genes A chimp has the exact same gene. Maybe it has different alleles, but it has the same gene. And you can tell because the sequence is very close. You share 92% of your genes with a mouse. You share 44% of your genes with a fruit fly. And 26% of your genes you share with a yeast. A yeast is not even an animal, it's a fungus. And a quarter of your genes are also found in that fungus. We actually use genetic homologies to create the tree of life. And if two organisms have genetic sequences that are really close to each other, then we know pretty much that they're closely related. If the organism sequences are more different, then we know that they are less closely related. Finally, the most convincing evidence for evolution of all is watching it happen in real time. So, speciation usually takes a long time to occur, but that doesn't mean it's impossible to see it happening. And right now, we actually have a speciation event occurring in killer whales in the Pacific Ocean. You have a couple different populations of killer whales in the Pacific, transient whales and resident whales. The resident whales will stay near a coastline, whereas the transient whales travel all across the whole ocean. And they actually have different food sources that they'll hunt. So the resident whales will live off of fish, whereas the transient whales hunt seals. And that difference in food sources means they have to have a difference in hunting strategies. Whales will communicate with each other with like clicks, and if you're hunting fish, then you can freely communicate with the other whales in your whale pod. But if you're hunting seals and you're clicking at the other whales, the seals hear that too, and they understand what it means. So they will get scared and like swim away. So when the transient whales are hunting their seals, they have to do it silently. For new baby whales, they learn how to hunt from the adult whales around them. So the best thing for a baby whale is if its parents share the same hunting strategy. And it can just learn like one strategy that works for a particular food source. So there's actually an advantage for these whales when they're trying to mate in seeking out another whale that belongs to the same basic population as them. Resident whales will try to be mating with other resident whales. Transient whales will try to mate with other transient whales. And they actually have different mating calls so that they can reliably attract mates of the correct type that they're looking for. So we call this reproductive isolation. You have these two populations of whales, resident and transient, and they are reproductively isolated from each other because they don't breed together naturally in the wild. They're using different mating calls to attract a whale of their same type, even if they sometimes share the same environment. Because they are reproductively isolated, when you have a mutation arise in one group of whales, it doesn't get transmitted to the other group, and they're becoming more and more different from each other genetically. And you can actually see some of those differences comparing the body of a resident whale to a transient whale. The transient whale as a whole is usually bigger than a resident whale. The shape of the dorsal fin is a little bit different. And you also have the white patch behind the dorsal fin that has a different shape in the resident and the transient whales. The whales are still able to mate, like physically they can do it and produce viable offspring, but they just don't choose to do that in the wild. But if you keep them in captivity, like they can mate with each other and create offspring. Also, like if you have a transient whale in captivity, and you try to feed it fish, it doesn't actually understand that fish are food. It will not eat them, and it will just starve. But if you keep it in captivity with a resident whale, and it gets to see the resident whale eating fish, then it understands like that those are food and it will start eating them. So at some point, the genetic differences between these two types of whales will be so large that they're actually not able to mate and create offspring together. At that point, they will be definitely different species. Another example of watching evolution happen is, unfortunately, antimicrobial resistance. So we've had antibiotics for, since like about the 1960s. So I guess about for 60 years. And they've really revolutionized medicine because now if you get sick with a bacteria and your immune system can't handle it, you can just take an antibiotic that will kill the bacteria. However, since we've been using antibiotics widely, we've started to see new strains of bacteria emerge that are resistant to our antibiotics. So this diagram shows how that process works. You start out with your population of bacteria. Most individuals in this population are susceptible to the antibiotic. That means that they can be killed by the antibiotic. They're vulnerable to it. Just by random chance, though, you have a couple individuals in this population that are resistant. So they have random mutations that give them resistance. As long as all you have is this population here by itself, they actually do not have an advantage. The resistant bacteria don't have an advantage compared to the normal bacteria. They actually have a small disadvantage because they're doing something for their resistance that isn't helpful to them unless there's actually antibiotics around. But they're spending some energy on it anyways, so they're wasting a little bit of energy right now. As long as there's no antibiotics applied, these guys are always going to be rare in the population. But as soon as you actually apply antibiotics, all these normal bacteria are starting to die, but the resistant bacteria don't die. And now all the food that was here, that all the bacteria were eating, is available pretty much all for just these couple of resistant bacteria. So they get all the resources for themselves, they start to multiply, and eventually they reestablish a full population. And this is now a brand new strain of antibiotic resistant bacteria. So the mutations that cause resistance are random, but using antibiotics is what gives bacteria that have those mutations an advantage. If antibiotics are used, it is inevitable that we are selecting for resistant bacteria. Those are the bacteria that have an advantage in any environment that includes antibiotics. So they're the ones that are going to have more offspring and pass on their genes. The graph at the top here is showing the rise of specific antibiotic resistant strains of bacteria. This is only a few of them. There's a lot now. The most famous one is this first line here. This is from MRSA. MRSA is methicillin resistant Staphylococcus aureus. Staph aureus is actually a skin bacteria. It's normal to have it on your skin and it's fine there, but if it were to get like deep inside a cut or something, then it can cause an infection. So sometimes people will get infections of it. And it used to be that we always treated those infections with methicillin, but in around 1975, we started to identify strains of Staph aureus that are resistant to methicillin, which is MRSA. And since that time, we have watched those strains become more and more common. This graph is only going up to like 2005, but already by that time, about 65% of all strains of Staph aureus that we found in the hospital were MRSA. They were resistant to methicillin. So now that most Staph aureus is resistant to methicillin, we have switched to using vancomycin for it instead. But now, of course, we're starting to see strains of MRSA that are also resistant to vancomycin. So not only are we seeing strains of bacteria that are resistant to a given drug, we're seeing strains that are resistant to multiple drugs. It's inevitable that this is going to happen as long as we are using antibiotics. The only way to, I mean, you can't really prevent it. The only way to slow it down is to use antibiotics less frequently. So for for a while, like once antibiotics came out, people just kind of regarded it as like a magic bullet, and they didn't really think about the fact that evolution exists and bacteria are going to evolve resistance. So they just prescribed antibiotics like willy-nilly for anything. And a lot of that behavior still even lasts today. Like, probably most of us have had the experience of going to a doctor because you're sick, and the doctor sends you home that day with a prescription for an antibiotic. That's not technically what's supposed to happen. First off, you shouldn't be giving antibiotics unless the person is like seriously sick. If it's like the immune system is probably going to be able to handle it, then the immune system should just be allowed to handle it. Second off, though, if you are going to prescribe antibiotics, you should know that they're going to work for the specific bacteria that the person is infected with. That means you need to find out what bacteria they're infected with, so you need to do some testing. And usually that's not done. Even today, it's still frequently not done. But this image at the bottom is showing that kind of test. You have a sample of the bacteria, which you would get maybe from swabbing the inside of a person's nose or their throat or something. Whatever tissue is infected, you would just swab it. And then you drag the swab across this petri dish and you let the bacteria grow. You put all these little white discs on there. Every one of these white discs is soaked in an antibiotic. And when you see that you incubate the plate and it ends up with a clear area around these antibiotics, that's because the bacteria are dead there because they can't survive in the presence of the antibiotic. So this strain of bacteria is killed by all these different antibiotics. On the other hand, this strain is a multi-drug resistant bacteria. It is killed by some of the antibiotics, but a lot of these discs don't have any clear area around them at all. The bacteria is fully resistant to those antibiotics. So now we'll talk a bit about the evolution of the flu virus specifically. Um, Mostly for new strains of the flu, we really care about just the H and the N proteins. Those are the proteins that the flu uses to get inside of a host cell. Because they're the surface proteins, they're also the easiest ones for the immune system to identify. So when the immune system learns what a flu strain looks like, usually it's learning what the H or N proteins look like and creating antibodies against them. So that means the H and N proteins are antigens. An antigen is just a molecule that the immune system can recognize and make antibodies against. The antibody is going to bind to only that one antigen and not any other molecule on Earth. And when it binds to its antigen, it directs the immune system to attack whatever is attached to the antigen. So when the immune system learns the H and N proteins, it will make antibodies against them, and those antibodies direct the immune system to attack the virus that those H and N proteins are attached to. Therefore, those are the proteins that we focus on when we make vaccines. And when the H and N proteins mutate, when they gain mutations and start to look different from the old versions, then it can become possible that the immune system doesn't recognize them anymore. So there's two trends in influenza evolution, which are antigenic drift and antigenic shift. In antigenic drift, you have normal mutations coming about in the H and N proteins, or I should say in the genes for those proteins, which create new alleles for those genes, and therefore new versions of the proteins. And if those new versions are different enough from the old versions, then the immune system can't recognize them anymore. So even though you have had the flu before, almost certainly, you're not immune to new strains of the flu that come out because they have brand new H and N proteins. The other thing that can happen is antigenic shift, which is where you still have the same alleles for the H and N proteins, but you have them in a new combination, and that can also make it harder for the immune system to recognize the virus. So antigenic drift is going to happen for any virus, any bacteria. It's just basically mutations happening, and they're always going to happen at some rate. But for influenza, because it's an RNA virus and it uses its own enzyme to copy its genome, and that enzyme makes a lot of mistakes, that means it has a lot of mutations. It has a high mutation rate. In fact, it's estimated that every single time the flu copies its genome, the enzyme that does it makes 2 to 3 mistakes. So every single time you have 2 to 3 mutations. That means every single flu particle is different from every other flu particle. Most of those mutations would be in other genes, not in genes for the H or the N proteins. That's only 2 genes out of all the genes a flu has. But, some of them inevitably will end up being in the gene for the H or the N protein and will create new alleles. Then antigenic shift. This is more of a rare phenomenon, but it can immediately create a brand new strain of the flu that has a new combination of H and N proteins. In order for antigenic shift to happen, you have to have one host cell infected with 2 different strains of the flu at the same time. So it's not good enough for the same animal to be infected with 2 strains at the same time. It needs to be literally the same host cell within that animal, infected at the same time with 2 different strains of the flu. So both those strains of the flu will be copying their genome, and you'll have new viral particles assembled. The genome of the flu is actually in 8 different pieces. Each new flu viral particle is going to have one of each of those pieces put inside of it. But if you had 2 different strains infecting the same cell at the same time, then when you're making the new viral particles, the 8 pieces are going to be randomly from one strain or the other strain. So at random, you could end up with the H protein from one strain and the N protein from the other strain, and right away you have a new strain. So, of course, every single year we have new flu vaccines coming out, and that's because we have new flu strains coming out every year. You always have antigenic drift going on, and some years we also have antigenic shift, ultimately creating new flu strains. The flu will infect humans. It can also infect pigs and birds, and it can infect ferrets. But for the evolution of the flu, what matters most is the fact that it infects pigs and birds including chickens. Because, of course, we eat chickens and pigs, that means that we raise large populations of chickens and pigs, and we keep them in really crowded environments with bad hygiene. So you pretty much are going to have multiple flu strains circulating in the pigs and in the chickens that we keep for our livestock. Most of those strains are not able to infect humans, but you just need a few mutations to switch a strain from preferring pigs or chickens to preferring humans. So inevitably, some of the strains that are infecting our pigs and chickens will evolve to infect humans every year. And we basically monitor which strains are circulating in our pigs and chickens and try to guess which ones are going to jump into humans. You have to try to guess which ones it's going to be, because if you wait till the strain actually jumps into humans, then you don't have enough time to mass produce a vaccine and distribute it before flu season. The way that we make the vaccine is kind of old school, and it takes months to make it, and you have to make like a lot of it as well. So they're always just kind of guessing which strains are going to be the strains that enter human populations. And of course, sometimes they end up being wrong, and then you end up with a flu vaccine that doesn't even like help with the specific flu strains that are circulating that year. But usually, actually, they're pretty good about predicting it. So earlier I said that the H1N1 flu strain can be transmitted really easily from person to person, but it usually doesn't cause a serious infection. The H5N1 strain is hard to transmit, but does cause serious infections and has a really high death rate because of that. The difference in those strains is what receptor they use. All flu strains will actually use a molecule called sialic acid as their receptor, but there's different versions of sialic acid on cells. And different strains of the flu will be able to recognize one version of sialic acid better than another. So that's the difference between those flu strains and their receptors. And you actually have different versions of sialic acid found in different places in the different species that are used as host by the flu. So for humans, flu strains that circulate well in humans usually use the alpha 2,6 version of the sialic acid receptor, which is found, you have a lot of it like high up in the respiratory tract and only a little bit down in the lungs. So a flu that prefers that receptor is going to easily find a host cell in the nose, but it's going to be hard for it to spread down into the lungs. For bird flu, the bird flu strains will prefer the alpha 2,3 version of sialic acid as their receptor. For birds, alpha 2,3 that's in blue here, it's found right there in their nose as well. So that kind of virus will transmit really easily in birds. But for humans, you only see the alpha 2,3 receptor deep in the lungs. So we have to breathe it in really deep to actually get infected by it. This means that bird flu in humans usually doesn't transmit well, but has a really high death rate, like 50% fatality or above. But it's super hard to catch it. And every time a bird flu jumps into humans, you have basically the possibility for a really high death rate. Usually that goes hand in hand with lower transmission though. So we've been able to escape like a real pandemic flu with a 50% death rate. But if you were to have a strain of the virus that can use both the alpha 2,3 and the alpha 2,6 receptor equally well, suddenly you would have a strain of the flu that transmits really easily and also has a high death rate. Then this is actually just showing the difference in those receptors. The alpha 2,3 sialic acid receptor is preferred in bird flu. The alpha 2,6 sialic acid receptor is preferred in humans. So here you have the sialic acid molecule, and it's always attached to a galactose sugar on the surface of a cell, and the specific attachment is different between alpha 2,3 versus the alpha 2,6. That's the only difference between those receptors. So now we'll talk about the functioning of the immune system. The immune system, of course, protects you against pathogens. A pathogen is anything that causes disease. It includes 3 lines of defense. The first line of defense is just your skin and your mucous membranes, which is like the lining inside your mouth and nose and stuff. You usually don't think about that much, but it physically keeps things out. Most things do not make it past the first line of defense. For anything that does make it past the first line, you have the second, which is your innate immune system. The innate immune system will respond to any pathogen or any cellular damage. It always has the same response to everything, which is just to cause inflammation. It can also cause fever, but its most basic response is just inflammation. It will react really quickly, but it's non-specific, so it treats all things the same. It can't recognize specific pathogens, and it responds the same way no matter what's going on. There's a lot of different cell types and a lot of different proteins that are part of the innate immune system, but the most important ones are macrophages, dendritic cells, and mast cells. and we'll talk about each of those in a bit. Then finally, you have the third line of defense, which is the most powerful part of the immune system. It's the adaptive immune system. This is the part of the immune system that recognizes specific pathogens based on their antigens and makes antibodies. It's really slow to activate, especially for the first time that you're seeing a given pathogen. But once it does activate, it's very powerful and it will create antibodies. Those antibodies will bind to the pathogen. Well, they'll bind the antigens on the pathogen, and it'll direct the rest of the immune system to target the pathogen more powerfully. The adaptive immune system is also able to remember pathogens that it has encountered in the past, or I should say, it remembers antigens that it has encountered in the past. So whenever you're gaining immunity to something, or if you have immunity to something, what that really means is the adaptive immune system knows it and recognizes it and remembers it. There's 3 cell types in the adaptive immune system, which are helper T cells, killer T cells, and B cells. The immune system overlaps extensively with the lymphatic system. So the lymphatic system is the network of lymph vessels that run alongside the blood vessels, and the lymph vessels will be catching fluid that leaks out of the blood into the tissues and returning it ultimately to the blood. Along the lymph vessels, you also have lymph nodes, which are full of immune cells. Lymph vessels are designed to be really easy to enter. So the idea is if you have bacteria in your tissue, those bacteria will start leaking into the lymph. The lymph will carry them to a lymph node, and there the immune system can see the bacteria and initiate a response. Another part of the lymphatic system is the spleen, which filters the blood and is also full of immune cells. So hopefully you would not end up with a pathogen in the blood because that's pretty serious right away. But if you do, at least the immune system can get a look at anything coming through in the blood. You also have immune cells in many other body organs. Most tissues actually include some kind of immune cell. In the blood itself, you have the white blood cells. Those are all different kinds of immune cells. Those cells are born in the bone marrow. Some of these cells for the adaptive immune system mature inside the thymus, which is an organ in the chest right above the heart. You also have a bunch of immune cells in the skin and the mucous membranes. Since those are lining parts of the body that are exposed to the outside, that's where most pathogens would be coming in at. So you always have immune cells there. And you also have some immune cells, like macrophages specifically, that just kind of wander around through tissues. Like they can migrate and they will walk through your tissues looking for pathogens. So we'll talk about the second line of defense, or the innate immune system, in more detail. We said there's macrophages, dendritic cells, and mast cells in that part of the immune system. The mast cells are the ones that are causing inflammation. Anytime they encounter evidence of a pathogen or any kind of cellular damage, they will activate. They will release cytokines. Those cytokines will cause inflammation. There's actually a lot of different cytokines that promote inflammation. One of them is histamine, and histamine in particular is really elevated in people who have allergies. So that's the reason why antihistamines are used as allergy drugs. But any kind of anti-inflammatory drug is always going to work by blocking one or more types of cytokines that are used by the immune system to promote inflammation. For macrophages and dendritic cells, they are phagocytes, which means a cell that eats things. They eat pathogens. Anything they eat is going to get destroyed. There's 4 cardinal signs and symptoms of inflammation, which are redness, heat, swelling, and pain. So if you recognize those signs, then you know that a particular body area is inflamed. The redness and the heat come from increased blood flow in the area. Inflammation will always dilate blood vessels in the inflamed area, which allows more blood to enter it. And the blood carries immune cells. So having increased blood flow brings in more immune cells and helps with the immune response. But then the blood is also red and hot, so it also causes redness and heat. Inflammation will also always cause the walls of the blood vessels to become more permeable, which means easier to cross. That way, the white blood cells coming into the area can easily leave the blood and enter the tissue. But that also means that fluid in the blood has an easier time leaving too, and that causes swelling. So that's why you have swelling with inflammation. Finally, the last sign of inflammation is pain. Inflammation causes your pain sensors to be sensitized, which basically means that stimuli that would normally not activate them will activate them. So normally, if you have like a light touch, that's not going to activate pain receptors. It activates just normal touch receptors. But if you were to bang your hand into something, that is enough force to activate pain receptors. But if your pain receptors are sensitized because of inflammation, even just a light touch can be enough to activate them, and that causes you to feel pain. The purpose of the pain is so that you will be aware that you're injured, so you can treat the injured part with care and let it heal. Inflammation is really helpful when it turns on and the problem gets resolved and then it turns off. But it can also be a problem in and of itself if it doesn't turn off or if you have way too much at one time. So, and also it does just cause like certain symptoms of sickness, like aches and pains. That's from inflammation. That's pain sensitization from inflammation. But if you have way too much inflammation at one time, it will lead to shock. Because inflammation always involves dilation of blood vessels, if you have way too much at one time, you'll have too many vessels dilating at once. That will drop your blood pressure. And if your blood pressure drops low enough that it can't sustain the flow of blood, then your organs are not receiving oxygen and you will enter shock. And either you'll receive medical treatment or you'll die. Then chronic inflammation is also bad. So you have some diseases that are just basically caused by inflammation. Like inflammation is the disease process. Anything that ends in itis would fall into that category. Itis means inflammation. So arthritis is inflammation of the joints. Hepatitis is inflammation of the liver, and so on. Allergies and asthma are also inflammatory diseases where you have just an overreactive immune system, including too much inflammation over time. So in all those conditions, you have ongoing inflammation, and that will always be damaging wherever it occurs. First, because it causes swelling, and an organ that is swollen usually can't work as well. But also because inflammation will always attract immune cells to the area. Those immune cells will come to kill things. If there's not bacteria for them to kill, they're just going to kill regular cells that are there. You also have inflammation playing a role in other chronic diseases like cardiovascular disease, diabetes, and even mental health conditions like depression. You can see elevated inflammation. There's a number of drugs that will reduce inflammation, but every drug has side effects. So there's no anti-inflammatory drug that is really safe to take necessarily like every day forever. Um, the most common ones are NSAIDs. NSAID stands for non-steroidal anti-inflammatory drug. Those are all your basic over-the-counter painkillers like ibuprofen, naproxen, acetaminophen, um, Tylenol, Aleve, all that stuff. That's all NSAIDs. Then you have corticosteroids, which are more powerful. They are the steroidal anti-inflammatory drugs. They work by blocking stress hormones. they work by mimicking stress hormones, and stress hormones block inflammation. Um, Both those categories of drugs, it's not safe to take for a long period of time. Like, they will cause side effects. The corticosteroids in particular can have really serious side effects. You also have montelukast, which is usually just prescribed for like asthma, but it also will block some inflammatory cytokines. Cannabinoids are anti-inflammatory as well, although they're not studied as well. And then, of course, exercise and diet. Exercise will lower inflammation, and having a good diet will also reduce inflammation. And those things do not have side effects as long as you're not exercising so much that you hurt yourself. The other possible thing that that the innate immune system can do is cause fever. Fever is caused by pyrogens. It's a class of chemical called pyrogens. Pyrogens can be released by immune cells, by damaged cells, or even by some pathogens. They will go to the brain. In the brain, the hypothalamus is the part of the brain that controls your body temperature. And whenever it's receiving pyrogens, it will just turn up the set point for your body temperature. And then your body just maintains the new temperature. We used to think that fever is like cooking out the pathogens, like they can't stand it being that hot. Actually, for most pathogens, that's not the case. Most pathogens are fine with a high temperature, and they actually have a much better tolerance of a wide range of temperatures than what you have, with a few exceptions. So probably the main thing fever is doing is just speeding up your immune system. All chemical reactions happen faster at a higher temperature. So if you raise the temperature, you raise the speed of chemical reactions in your immune system. Fever doesn't feel good. It causes like chills and malaise. Malaise is just a general feeling, like a general bad feeling. If it gets more serious, it can cause like hallucinations as well. And a very bad fever will actually put you in a coma and cause brain damage and can kill you. Although that would only be possible with like an extremely high fever, like 108. So, in general, you don't need to worry about like treating a fever. In fact, it's better if you don't. There are some over-the-counter drugs that reduce fever, like Tylenol. But actually, studies show that when people take Tylenol for their fever, they actually end up being sick for longer than if they wouldn't have taken anything at all. So the fever does help you get over the sickness, as long as it's not way too high. If your fever's like 103, 104, you don't need to worry about it. You should probably go to the hospital though before it actually gets to 108. So like, like 106, start to worry. Then the third line of defense, the adaptive immune system. This has three types of cells: the helper T cells, the killer T cells, and the B cells. Killer T cells, like the name suggests, they kill. B cells make B cells make antibodies, and helper T cells are used to turn on the other two cells. Every cell in the adaptive immune system is able to recognize antigens. So it will have a receptor on it that basically looks like an antibody and can only bind one particular antigen. In order to turn the adaptive immune system on, it's a really complicated process. And I'm not going to really put that on the test, but we will like go over it. So first you have to activate the innate immune system in order to turn on the adaptive immune system. The cells of the adaptive immune system can recognize antigens, but they're not going to turn on just because they see an antigen that they recognize. They need to be shown that antigen in a specific way. And the first kind of cell you need to activate is the helper T cell, because that's the one that's going to turn on everything else. Or I should say, help turn on everything else. So in order to activate a helper T cell, you have to have a macrophage or a dendritic cell eat the pathogen and show the antigens from that pathogen to the helper T cell. So macrophages and dendritic cells, we said they're phagocytes, so they will eat pathogens. Anything they eat, they're going to chop it up into little bits, and they will actually put those bits on the cell surface. They will display it on their surface. Then they will go look for a helper T cell and try to show it the antigen. And every helper T cell has one specific antigen that it can recognize. So the macrophage or the dendritic cell needs to go through a bunch of different helper T cells until it finds the one cell that can actually recognize the antigen that it's showing and activate. At that point, they will bind together. The helper T cell and the macrophage or dendritic cell will bind together at the antigen, and the helper T cell will turn on. Once the helper T cell is turned on, it's going to start dividing, and it's going to release cytokines. Those cytokines are needed to turn on the killer T cells and the B cells. Killer T cells don't kill pathogens directly, actually. They kill infected cells. So, for instance, a cell infected with a virus, a killer T cell would be able to identify that cell and kill it. The goal is to kill the infected cell before the virus can complete its life cycle within that cell. So whenever a cell is infected by a virus, it is actually going to end up showing little pieces of the virus on its surface. So it will show antigens of the virus on its surface, and the killer T cell can come by and see that. But the killer T cell will not activate just because it sees the antigen it recognizes on the surface of an infected cell. It will only activate if it also is receiving cytokines from a helper T cell at the same time. So you have to activate the helper T cells first, then you can activate killer T cells. Once a killer T cell activates, it again is going to start dividing and will go around looking for any cell infected with the pathogen that has the antigens it recognizes, and it will kill that cell when it finds it. The last kind of cell in the adaptive immune system is the B cell, which is the one that makes antibodies. B cells will actually bind to their antigen directly on a pathogen, but again, they're not going to actually activate unless they bind to the antigen while they're also receiving cytokines from an activated helper T cell. Once the B cell turns on, it will start dividing and making antibodies. Then the antibodies will also be binding. They'll be binding to the antigen wherever it is, and they will direct the rest of the immune system to attack at that location. There's actually different kinds of antibodies and a lot of different functions that they have, but they will help other elements of the immune system identify the antigen or identify the pathogen that is attached to that antigen and attack it. They can also clump pathogens together, which makes it harder for them to cause problems in your body. And if you have enough antibodies, they can actually completely coat the surface of a pathogen. If you have like a virus and it's completely coated by antibodies on its surface, then actually it's not able to grab a host cell. So you're basically, in a way, destroying that virus just by coating it with antibodies. Whenever any cell in the adaptive immune system activates, it's going to start dividing. Some of the cells that it makes are going to be involved in like killing infected cells or making antibodies, depending on what the cell type is. But some of those new cells will be memory cells. So whenever a helper T cell, a killer T cell, or a B cell activates, it's going to divide, and some of the new cells will be memory cells. A memory cell is not active right now. Its only job is to live a long time and remember what the pathogen looks like or what that antigen looks like. This is the basis for your immune system's ability to remember things from the past. When you are infected with a pathogen for the second time, you have memory cells for the antigens of that pathogen. The memory cells will activate really quickly and easily, and you'll have a really fast, strong adaptive immune response. So when a memory cell is turning on, you don't have to go through a long, complicated process. You don't have to have a helper T cell activate first. It doesn't have to be shown the antigen in any special way. When a memory cell sees an antigen in any context, an antigen that it recognizes, it will turn on and activate. This means that the second time you're exposed to a pathogen, you have a much faster adaptive immune response, which makes you effectively immune to that pathogen. So for the adaptive immune system, it has primary and secondary responses. The primary response is what happens the first time you encounter a pathogen. The secondary immune response happens like the second time or the third time or any other time that you encounter that same pathogen. The primary immune response is slow and kind of weak. The secondary immune response is fast and very strong. In the primary immune response, you're making your memory cells. If you have memory cells for this pathogen, then you can have a secondary immune response. So the secondary response requires the memory cells. For your primary immune response, it can take like half a week to start making antibodies against the pathogen that you're sick with. And then the total amount of antibodies that you make is not going to be like that high. For the secondary immune response, you can be making antibodies the first day that you have the infection, and the amount of antibodies that you make is going to be a lot higher overall. So it's a faster and a stronger immune response. Another or a final point about the adaptive immune system is that it's very important that when the adaptive immune system is developing, it learns the difference between self and non-self. Every single cell has antigens. That includes your own cells. An antigen is any molecule that is sufficiently complicated in its shape that the immune system can recognize it specifically and make an antibody against it specifically. That includes plenty of molecules that are found on your own cells. When you're born, you have a huge population of helper T cells, killer T cells, and B cells that are recognizing all kinds of antigens, just totally at random, because you can't like predict which pathogens you're going to see in your life. So you'll be born with a population of adaptive immune cells that can recognize millions of different possible antigens, just at random. Some of those antigens inevitably are actually found on the surface of your own cells. So any adaptive immune system cell has to go through a process of maturation. And during that process, the most important thing that happens is that any cells that are responding to antigens that are part of your own body are identified and killed. The consequences of that not happening right are that you end up with mature T cells or B cells that can recognize one of your body's antigens as a foreign invader, basically, and activate in response to it and kill it. Um, that's going to result in an autoimmune disease. An autoimmune disease is a disease where the immune system is attacking part of your own body as though it was a pathogen. So that's based on the immune system recognizing an antigen that's part of your normal cells or part of your body. For B cells, they go through the maturation process in the bone marrow. For T cells, they go through that process in the thymus. And the thymus is an unusual organ because it actually gets smaller as you age. When you're a kid, you have like plenty of thymus, but when you're an adult, most of it's gone. Most of it's just turned into fat. So that means that the T cells that you mature as a child are supposed to last you for your whole life. When you're an adult, you have only like a small ability to mature new T cells. And that comes into play in like HIV. The HIV virus infects helper T cells. That's its host cell. And you need helper T cells to turn on all the other cells in the adaptive immune system. So when you have the virus, it's going to start slowly over time killing your helper T cells. Eventually, when all your helper T cells are dead, at that point you have AIDS. and you're immunocompromised. Because you don't have helper T cells, you're not able to turn on your remaining killer T cells or B cells, and because most of your thymus is just fat as an adult, you're not really able to replace the the helper T cells that are gone. And the last thing we'll talk about in this unit is vaccines. So the goal of a vaccine is to teach your immune system what a pathogen looks like so it can recognize it in the future. Basically, what this means is the vaccine needs to cause a primary adaptive immune response. So in order to do that, it needs to have at least an antigen from the pathogen, and it needs to activate the innate immune system. That way, you can go through the whole process of having a macrophage or a dendritic cell show the antigen to a helper T cell and activate it, and then activate the killer Ts and the B cells. So the vaccine needs at least an antigen from the pathogen and the ability to activate the innate immune system. Then when you get the vaccine, you go through a primary adaptive immune response. You generate memory cells that remember that antigen that was in the vaccine. And then if you see the pathogen that normally has the antigen in real life, you already have memory cells for the antigen. So you get to have a very strong, fast secondary immune response. That immunity is going to last as long as the memory cells are alive. And they are long-lived cells, but sometimes they do die, so it's actually kind of common for a vaccine that eventually you would need like boosters to re-up your immunity. For viruses in particular, vaccines are the best thing we have. We don't really have drugs for viruses the way we have antibiotics for bacteria. There are some antiviral drugs, but there's not nearly as many of them as there are antibiotics. And that's because a virus is very simple compared to a bacteria. A bacteria is a full-on cell that has a metabolism and the ability to reproduce and everything. It has a ton of enzymes that are serving all those functions, and if you can block one of those enzymes, you can take out a crucial function of the cell that would be fatal for that bacteria. But for a virus, they don't have that stuff. They don't have a metabolism or a reproduction. Instead, they're pretty much stealing from the host, and you can't really interfere with the functioning of the host cell because that's your cell. So, sometimes it is possible to make antivirals for a virus. For viruses that have their own specialized enzymes, sometimes you can make a drug that blocks the function of that enzyme, but usually there's just not really much to target there. That means that we really just rely on the immune system for defeating viral infections. And you saw that during COVID, especially in the early part of the pandemic, we really had no drugs at all. And so what we had is pretty much trying to keep the person alive while their immune system clears out the infection. So the infection is so bad that you get fluid in your lungs and you can't breathe anymore. We put you on a ventilator so that you can survive and just wait for your immune system to clear the virus. The other thing we have is vaccines. So with a vaccine, you can prevent yourself from becoming seriously ill in the first place because your immune system already knows what this virus looks like and can respond to it very quickly and strongly. Typically, a vaccine is not going to prevent you from getting sick at all. Like, it won't make it so that it's impossible for the virus to enter your body and find a host cell, usually, but it means that you're not going to have a serious illness because the immune system is going to be responding so quickly and so heavily to the virus. The other nice thing about a vaccine is that if you have enough vaccination in a population, you can eliminate a disease from the population. And we've done this with smallpox and to some extent with polio. So smallpox in particular used to be a scourge of our species. Um, Like, kings throughout time have been frightened of smallpox. But smallpox no longer exists because we made a vaccine for it. Essentially, every person on Earth took that vaccine, and smallpox could not find a host, so it died out. It exists now only in vials in a lab. The other disease that we have tried to eliminate is polio, and that one we've had um, some, like, I guess, less success with. So the original polio is gone, but there is still polio in some parts of the world today. The polio that exists today is actually viruses that are descended from the vaccine because of the type of vaccine that it was. So the earliest polio vaccine was actually just the polio virus, but like a weakened version of it that can't cause polio. But it was still like a functioning virus. So it was still replicating in host cells, and that means that it can evolve. And in some cases, the vaccine virus did actually evolve to regain the ability to cause polio. And so that's the polio virus that circulates today is from the vaccine virus. New polio vaccines are not like that. They don't have an actual live virus in them. But there are actually some parts of the world where they're still using the original vaccine just because it's cheaper. So this table is showing the reduction in disease rates thanks to vaccines. And you can see smallpox down to 0, polio down to 0 with a little star. The star is because of the vaccine descended virus that is now causing infections in some parts of the world. But in like the first world, of course, it's eliminated. Even for diseases that have not been actually eliminated, like the measles, the cases have been reduced massively because of widespread vaccination. Although we are starting to see some measles outbreaks again because people are not vaccinating. Some people are more scared of the vaccine than they are scared of the viruses that the vaccine protects against. So this is some of the types of vaccines. Um, the most dangerous type is the attenuated vaccine. That is the kind of vaccine that like the first polio vaccine was. So it's a vaccine that has a live virus in it. It is attenuated, which means weakened basically, so that it can't cause disease, but it's still like a functioning pathogen. It can still replicate. That means it can still evolve. So that makes it the most dangerous kind of vaccine, and we don't really use attenuated vaccines anymore. The plus side is, though, that because the vaccine contains an actual living virus that can replicate in host cells, you actually have a really strong immune response to this kind of vaccine, and you get usually like immunity that lasts for your whole life. So the best immunity possible is from an attenuated vaccine, although it's also the most dangerous kind of vaccine. Um, After they came out with attenuated vaccines, they came out with inactivated vaccines, where the vaccine still has a virus, but it's killed. It's not able to enter host cells or replicate in host cells at all, which means it's a lot safer, but also it's not going to give you as good immunity. It still gives pretty good immunity though, because the vaccine contains like the whole virus. Those are both pretty old categories of vaccine that we've had for a long time. Newer vaccines would be subunit vaccines and mRNA vaccines. The subunit vaccine is a kind of vaccine that doesn't have the whole virus. It only has the antigen protein. That means it's very safe because, there's just no viruses involved. Um, but it doesn't give as good immunity because it's only one antigen from the virus. So like for the flu, it would be like just the H protein or just the N protein maybe. So we'll probably need boosters for that kind of vaccine. And the newest kind of vaccine is the mRNA vaccine, which didn't exist before COVID. This kind of vaccine contains mRNA for building the antigen. So for like the COVID mRNA vaccines, COVID had the spike protein on its surface that would bind to host cells and intra-host cells. And in the mRNA vaccines, they included the mRNA for the spike protein. So you introduce that mRNA into your cells will use the mRNA to build protein off of it. So they're going to start building the spike protein, and they put it on the cell surface, and then your immune system can come by and see it and have a response. This is also a very safe kind of vaccine, but like the subunit vaccine, it doesn't give like fabulous immunity, so you would probably need boosters. The benefit of an mRNA vaccine over a subunit vaccine is that it's a lot faster to produce it. In a subunit vaccine, you have to produce actual protein. That takes a lot longer and is a lot harder for us to do compared to making mRNA, which we are really good at. So vaccines are overall very safe, but they're not like perfectly safe. Um, First, like, it's pretty common to have some symptoms after you've been vaccinated that would be looking like symptoms of sickness, because the vaccine is turning on your immune system, and because the immune system causes most symptoms of illness. That means that you may have some feeling of sickness. That is just a symptom of your immune system turning on. You're not actually sick. It's also possible to be allergic to something in a vaccine, which is why they tell you to wait around for like 10 minutes once you get a vaccine. Usually, if you're going to be allergic to it, you're going to have that reaction pretty quick. So if you're still in the area where you got the vaccine, they can give you an epi shot. For the older kinds of vaccines, the attenuated vaccines in particular, those are dangerous because the virus they have can evolve. For the inactivated vaccine, like, the virus in there is dead, but it's still possible that it could be alive, because, like, you try to kill it, but what if you tried to kill it and it didn't work, but you didn't realize that it didn't work? Now you have a dangerous product. So with those kind of vaccines, it is always possible for there to be some danger involved. For the subunit and mRNA vaccines, though, that's not possible because they just don't contain a virus at all. If you are immunocompromised or if you're pregnant, then it's generally less advisable to receive a vaccine. Vaccines only work by teaching your immune system how to recognize a pathogen. So if your immune system is like broken anyways, then it's not going to work as well for you. If you're pregnant, then your fetus is immunocompromised. Um, Even with all this, it is still possible for like serious reactions to a vaccine to occur, and we don't really necessarily always understand why that is. So for like the smallpox vaccine, about 1 in 1 million people had just a really serious bad response to the vaccine and they died. Um, that's of course like a very, very small death rate, especially compared to the actual disease of smallpox, which has killed legions of people throughout time. But it still sucks to be that one. For the COVID vaccines, some of them did cause like heart issues, which again were very rare, but some people do have a bad reaction. Overall, vaccines have very high safety standards. For any medication, you always judge the safety of it by looking at like, what are the consequences of not treating this person versus what are the possible side effects and consequences of using this treatment. But for a vaccine, you're giving that to a person who's actually not sick. So you have to weigh the consequences of them, or like the chances that they would get sick, and then the possible consequences of them being sick versus the possible side effects of the vaccine. So the bar for vaccine safety is set a lot higher than it is for different kinds of medications. There are myths about the safety of vaccines. The oldest, most persistent one is that vaccines cause autism, and it was specifically like the MMR vaccine that was accused of causing autism. This is not accurate. This myth originates from a guy, Andrew Wakefield, who was a doctor. He published a paper in 1998 claiming to show a link between this vaccine and the development of autism in a patient population he was working with. It turns out that he falsified that data. He was actually paid almost $500,000 by some lawyers trying to sue the vaccine manufacturers to falsify the data. So he was stripped of his medical license for that. But unfortunately, the myth does persist. So the MMR vaccine is the one that gives immunity to measles. Um, Vaccination rates with that vaccine have dropped because, in large part, of this belief, which is leading to outbreaks of measles now. And we actually had one in the New Mexico and West Texas area not too long ago, which did have some deaths. And people who die from measles usually are like little kids. Yeah, so that's unfortunate. Since his paper was published, scientists have actually gone and exhaustively investigated any possible link between autism and the MMR vaccine. There is no link.